In blood, there are three types of form elements: RBC, WBC, and platelets. Platelets are also known as thrombocytes. They are known as thrombocytes. Means thrombus means clot and this cytes means cell. This is thrombocyte. Size size of cell is 2 to 4 micron. 2 to 4 micron in diameter, and they are colored as oval rod-shaped biconvex structure without nucleus. There is no nucleus. No nucleus. Mitochondria, Golgi bodies, rough endoplasmic reticulum, glycogen storage are present, but nucleus is not present. This is size, color, and normal count is 1.5 lakhs to 4 lakh per microliter. When it is less than 1.5 lakhs, then it is known as thrombocytopenia. Thrombocytopenia. Less than 1.5 lakh per microliter. And when it is more than thrombocytosis. When more than 4 lakh per microliter. Okay. Now, the structure of thrombocytes. The structure of thrombocyte. This cell membrane is there, and in cytoplasm is there. In cell membrane is covered by glycoprotein coat with receptors GP, glycoprotein 1b, glycoprotein 2b, and glycoprotein 3a. This cell membrane, in cell membrane, protein 55%, fat 42%, and carbohydrate 3%. This is the composition. Fat, phospholipid. Phospholipids are converted into arachidonic acid by phospholipase A2, and this is converted into prostaglandins and leukotrienes. These substances are essential for clot formation. They accelerate clot formation. Glycoprotein prevents adhesion of platelet to normal endothelium. Function of glycoprotein is prevention of adhesion of platelet to normal endothelium, but it increases adhesion in damaged endothelium collagen fibers. Okay, these are the functions. Now cytoplasm. In cytoplasm, actin, myosin, thromboasthenin, rough endoplasmic reticulum, Golgi apparatus, mitochondria are present, and fibrillin stabilizing factor is also there. Now it contains three types of granules. Alpha granules, dense granules, and lambda granules. Alpha granules, dense granules, and lambda granules. Three types of granules are there. It is clear? In cytoplasm, three types of granules are present. Alpha granules, dense granules and lambda granules. Alpha granules contain protein substances, non-protein and lysosomes. Okay, in alpha granules contain protein molecules, protein substances, variety of coagulation factors are there. Variety of coagulation factor is there. Fibrinogen, von Willebrand factor, platelet derived growth factor and platelet factor 4. Vascular endothelial growth factor, endostatin and thrombospondin are present in alpha granules. Okay, and in dense granules, non-protein substances like ATP, calcium, serotonin, and lambda granules, lysosomes. In lambda granules, lysosomes with acid hydrolases and other enzymes are there. When activated, when cell is activated, platelet is activated, granules are discharge their contents and promote platelet adhesion, aggregation and fibrin deposition. When activated, when platelet is activated by ADP or calcium ion, it forms pseudopodia-like structure. This is activated platelet. And when activated, then these granules, dense granules, alpha granules, lambda granules discharge their content and promotes platelet adhesion, aggregation and fibrin deposition on the platelet. Okay. Now properties of platelets. Properties of platelet: adhesiveness, aggregation and agglutination. Adhesiveness, aggregation and agglutination. What is adhesion? Adhesion is when this is blood vessel. Now when it is damaged, when blood vessel is endothelial layer is damaged, then collagen is exposed. Now collagen is exposed, bloods and platelets are moving in the blood. Platelets are moving in the blood. They are attracted by the collagen fibers and lamina and they come here. They become activated. They become activated. This is adhesiveness. This is accelerated by ADP, von Willebrand's factor secreted by the endothelial collagen fibers, and this causes adhesion. Adhesion means attraction between two different things. Here it is collagen fiber and this is platelet. Then adhesiveness. It gets adhered here to the collagen fibers. Now after this, activated platelets will form pseudopodia. Pseudopodia like a structure. The pseudopodia like a structure will spread from here and this will activate another platelet and they become activated and this is aggregation. Aggregation between same things. Two platelets, three platelets aggregate to form the plug and they pile upon each other. Agglutination. They pile upon each other. These are the properties of platelets. Okay, these are the properties of platelets. Now formation of platelet or thrombocyte. Formation of or synthesis of platelet is known as thrombopoiesis. Process is thrombopoiesis. Thrombopoiesis is also known as megakaryocytopoiesis. Megakaryocytopoiesis. That is the pluripotent stem cells present in the bone marrow is stimulated by interleukin 1, interleukin 6, interleukin 3 and granulocyte macrophage colony stimulating factor to form committed stem cell. Pluripotent stem cell to committed stem cell to megakaryoblast. From megakaryoblast, it is stimulated by granulocyte macrophage colony stimulating factor that will form pre-megakaryocyte, pro-megakaryocyte, and granular megakaryocyte. Granular megakaryocyte granular megakaryocyte, when passing through the sinusoids, they are large cells, and when they try to pass through this sinusoids of blood vessel, then one fragment is get detached. One fragment get detached, then another fragment get detached, and they are known as platelets. Fragments of cytoplasm. This is platelet. This thrombopoiesis is regulated by colony stimulating factor. Colony stimulating factor plus thrombopoietic stimulating factor. Two things stimulated, regulated by colony stimulating factor and thrombopoietic stimulating factor. And thrombopoietin is secreted by liver and kidneys. When thrombopoietin, like erythropoietin, thrombopoietin, thrombopoietin is secreted from liver and kidneys. This thrombopoietin stimulates, increases formation of platelets. Formation of platelet. When excessive platelet destruction occurs, this will increase formation of thrombopoietin and hence formation of platelet. This is feedback mechanism. In liver diseases, thrombopoietin is reduced because it is formed mainly in the liver. It reduces and therefore platelet count also reduces. Okay, in decreased platelet count, I said it now, decreased platelet count due to destruction will stimulate formation of thrombopoietin formation, and this will increase platelets. Number two, in fever, interleukin 6 increases. In fever or infection, inflammation, in infection, interleukin 6 increases, that will increase TPO, that is thrombopoietin, that will increase platelet count. Interleukin 6 increases formation of TPO and so increase in platelet count. This is about formation of thrombopoiesis. Now, functions of platelet. Number one, hemostasis. It forms hemostatic plug and maintains the integrity of hemostasis. Forms clot retraction. It is clot retraction. Third is phagocytosis of carbon particles, viruses, and immune complexes. Okay. Number 4, storage. Number 5, transport, storage of 5-hydroxytryptamine, and that is histamine, and transport of this 5-hydroxytryptamine and histamine. Number 6, repair of damaged blood vessels, repair of damaged blood vessels, and number 7, vasoconstriction. Vasoconstriction by this releasing histamine and other. These are the functions of platelet. It maintains the normal vasculature. It is responsible for bleeding time. How you will know the number of platelet? Just by complete blood count, CBC, or by slide test, peripheral blood smear, we can know the number of platelet. And if it is less than normal, that is 1.5 lakh, this is thrombocytopenia. It is destroyed in spleen. Therefore, when function of spleen increases in splenomegaly, platelet count will decrease, but never less than 40,000 per cubic milliliter. Okay, thank you. Thank you to visit. Thank you for watching. If you get something from this video, like it, share it, and subscribe this channel. Okay, thank you. Thank you very much.